The overall goal of this procedure is to provide a simplified system for the analysis of cell Durotaxis, through which many mammalian cells preferentially migrate towards a more rigid substrate. This is accomplished by first preparing polydimethylsiloxane, or PDMS, substrates of defined rigidity. Next, the prepared PDMS is poured into each well of a 6-well plate, and any air bubbles are removed. The Durotaxis chambers are then completed by placement of a sterilized cover slip on top of the PDMS in each well to create an overlay. The final step is to seed cells into the Durotaxis chambers at a density that gives the cells enough room to migrate without significant interactions with other cells. Ultimately, live cell microscopy is used to study Durotaxis in migrating cells.
The main advantage of this technique over existing methods, like polyacrylamide, is that there is a single step between the soft PDMS and rigid glass instead of gradient gel alternatives. Also, coating PDMS with extracellular matrix components, like fibronectin, do not require chemical cross-linking, which is required for polyacrylamide gels.
To prepare one 6-well tissue culture plate, first tare the balance with a 50 milliliter conical tube. Weigh out approximately 10 grams of the polydimethylsiloxane, or PDMS, base solution in the 50 milliliter tube. For a 90:1 substrate, divide the measured weight of the PDMS base solution by 90.